I'm holding here what we call a mini rhizotron tube. It's a clear plexiglass tube that we insert into the soil at an angle down to about a meter in vertical depth. And we insert a specialized camera for imaging the roots, and from that we can track the root growth over time and convert that root growth into carbon sequestration and the amount of carbon that's sequestered below ground. I want to talk a little bit about how we measure carbon sequestration in the forest, the many tools that we use to quantify the amount of carbon that goes into the forest and where that carbon ends up once it's in the forest and sequestered in biomass and soils. So we use a variety of tools. We use a meteorological or carbon flux tower, and that flux tower measures the exchange of carbon dioxide between the forest and the atmosphere about every half hour, so on a half-hourly time step, which is a really relatively high frequency measurement. It's telling us how much carbon is entering the ecosystem as a whole and how much is occasionally being lost. For example, at night, when the sun goes down, carbon is emitted. During the dormant season, when the leaves are off the trees, carbon is being emitted. But this time of the year, we have green leaves on the trees in our deciduous forest. Carbon, for the most part, is going in. There's a net carbon sink associated with this forest. That we can measure, again, with fairly high accuracy using a meteorological flux tower. Meteorological flux towers are fairly expensive, and they don't tell us where that carbon is once it enters the ecosystem. So we employ a variety of tools, many of which are behind me in this forested ecosystem, to quantify where that carbon is in the ecosystem and how much of it is located in a given pool within an ecosystem. We also look at dead wood pools. So I mentioned that facet resulted in the death of tons of carbon actually worth of live trees. That carbon ended up, much of it, on the forest floor, and it's still here over a decade later. So that's an important pool that we can quantify simply by using tape measures and scaling the volume up to carbon mass. We also have dendrometer bands on trees. These are bands almost like a belt around a tree that measures the radial change in growth of a tree. From that radial growth, we can translate that incremental growth into wood biomass, and then we can take wood biomass and translate that into carbon and estimate how much carbon is sequestered in the trunks of trees. We can also estimate the amount of production and carbon produced and allocated to leaves on an annual basis. And we do that through litter traps. They're simply mesh screen bags essentially that we keep out in the forest that gather the litter. We take that back in the lab, we dry it, we weigh it. And because just about everything you see here in the way of biomass is about 50% carbon, we can pretty easily translate that biomass into a carbon mass. So that allows us to make a comparison and in principle take the data from the tower, and if we know how much carbon is going into the ecosystem, we should be able to find it all through those various measurements that I described. I would just add one other thing that's really interesting about this system and our forests in general is about half of our carbon is above ground and invested in biomass pools, so the living and dead vegetation that you see above ground, but that's matched below ground, which is pretty incredible when you think about it. So consider that all the live vegetation around us, if we dried that vegetation, weighed it, 50% of that dry weight mass is carbon. That's equal below ground. So the carbon pools below ground are also really incredibly important for us to understand, to calculate, to quantify and characterize as they change over time in response to this experiment. All right, we're here at the base of the Forest Accelerated Succession Experiment Tower. This particular tower is 34 meters tall, almost 100 feet, and looking up, what I can see are sensors that are pulling carbon dioxide, pulling air really, through tubing that's then running through a pump house over here, and then running through gas analyzers and telling us at a very high frequency how much carbon is being exchanged between the atmosphere and this forest. The range of area of measurement varies a little bit depending on the wind speed and the wind direction, which we cannot control. So that means there's a floating dynamic footprint, we would call it, or area of measurement. Most of the time that's concentrated within the area of manipulation where we stem girdled all the aspen and birch to create this disturbance experiment. Occasionally it floats to the west, occasionally it floats to the east or north, and that depends again a little bit on the origin of the fluxes, which is dependent on the wind direction primarily. For this approach to work, it's a passive approach, so carbon dioxide is being passively exchanged using the Eddy covariance technique. So this is the exchange of carbon dioxide between the canopy and the atmosphere. I like to think of it as very much like eddies rolling over a rock and being produced and kicking up sediment in a stream bed. That same principle applies to how carbon dioxide is exchanging with the atmosphere. Air is moving across a rough canopy, and just like that rock, it's kicking up CO2 up and down, rolling over the top, the surface of this canopy. And that's allowing us, in fact, to take these measurements without having to generate our own suction or wind patterns to bring in an active way the carbon from the forest. It's all passive exchange using that turbulence or wind. This particular forest sequesters on average about 2 to 3 tons of carbon per hectare per year. That's metric tons. That's about as much as your average vehicle will emit in a year. Typically, some vehicles will average a little bit more, a little bit less, but that gives you some perspective on how much carbon is being sequestered by this forest annually. And again, we can measure that very precisely using this tower with high accuracy. What we can't do is tell you exactly where that carbon ends up once it enters the forest.
So what I'm working with is a Lycor 6400, and what I have attached here is called an IRGA, an infrared gas analyzer. And what this little chamber does, right now I'm just calibrating it. It's not attached or taking any measurements. It's just trying to figure out like where the zero is. And the zero we're referring to is like zero CO2 in the chamber kind of deal. So there's a bunch of tubes connected here, and we have some soda lime and some dryerite, which are taking all the water and carbon out of this little chamber. Once we zero it, we can know how much gas is in the chamber, and what I'm looking for is going to be soil respiration rates. And so this chamber will basically put it to a known level of CO2, which right now we do about 400 parts per million because that's similar to atmospheric levels. And then once it hits that 400 ppm, it starts measuring and recording from these little chambers, and that gas builds up in the chamber, and you can use, the volume inside this chamber, how much gas builds up over a certain amount of time to find a rate of carbon accumulation. That's what we call our efflux. The process is soil respiration, which is kind of the counterpart of primary production in the global carbon cycle. So primary production is taking carbon out of the atmosphere, soil respiration, releasing carbon from the forest back into the atmosphere, and that's coming from, this plant material decomposing on the ground. Also, roots are respiring underground, fungus, all those things that are, just breaking down and decomposing, that's releasing the carbon, and that's what we're measuring in here. Theoretically, yes, there is, a younger forest has less detritus, less leaves and things that have fallen to the ground. And so there's less respiration going on, less decomposition. And as stands age, more of that builds up and more decomposition is therefore happening. A lot of our work and I guess, theoretically, at some point that respiration surpasses that primary production that I was talking about, and the forest as a whole is releasing carbon rather than taking it in. But a lot of work by our lab has kind of found things contrary to that, because different disturbances can come in and kind of shift where a forest is at. Sometimes they are resilient and they bounce back to previous levels. Sometimes other things change. So, like I said, the chamber kind of records amount of soil respiration per volume. And we take that at 5 different areas across the plot and average it. So we're saying like the average of this stand of this forest soil respiration is this number, and we scale it up to the space that the plot takes and then up from there. So the data that we collect is really informing models. So soil respiration is one part, primary production is another part. So if somebody's taking data from tree cores or from leaf litter to figure out, different pools across the forest, you can take these small measurements, use some math, scale it up, and kind of add and subtract it all together. So like you're adding all the production values and subtracting all the respiration values, and that can give you one number of like, okay, so this forest either annually or over a set amount of time has stored this much carbon or is releasing this much carbon, and that kind